The overall goal of the following experiment is to quantitatively measure the subnuclear organisation in yeast cells and how it is affected by changes in growth conditions or mutations. This is achieved by growing yeast cells to log phase in conditions that will minimise autofluorescence. Next, the cells are placed into a growth chamber for viewing under the microscope. Then, images are taken to measure the distance between different fluorescently labelled structures in the cell. Results are obtained that show differences in subnuclear organisation, which is dependent on changing the nutritional conditions during growth of the yeast culture.
This method can help answer key questions in the epigenetics field about the interplay between subnuclear organization of chromatin and gene regulation.
To culture fission yeast, begin by preparing Edinburgh minimal medium or EMM and EMM without ammonium chloride. To reduce autofluorescence, rather than autoclave, use a 0.2 micron filter to sterilize the glucose solution and add it to the autoclave media. Inoculate 3 ml of EMM in 30 ml tubes with fission yeast cells freshly grown on an agar plate in the rich medium YEA. Lightly cap the tubes and incubate the cells in a shaker at 225 rpm and 30 degrees Celsius. Keep the cells in log phase for 2 days by counting them with a Birka chamber every morning and evening and diluting them to the appropriate density. On the day of the experiment, the cells should be in early log phase. To nitrogen starve the cells, refer